Hello, good afternoon, everyone. I hope you can hear me. Yeah. Hello, Ira. I hope you can hear me and uh, the volume is correct. Okay, great. Hello. Okay, so we will start the session uh, right now because there might be a bit. I mean, there is not much to cover, but there are steps in which Galaxy will take time. So I guess we can start right now. Yeah. So thanks for coming for the second session. And um, just a quick reminder that today we'll be talking about RNA-Seq analysis, but mostly about how to get the count stable and how to at least start doing the analysis, and how to control for covariates. So how to control for factors in RNA-Seq that you do not want to, you're not interested in. So something like batch effect and these kind of things. Yeah. And just a quick reminder that this is the GitHub page. Yeah, I think you all know. And I did not update it, but now Yura has also put notes from the previous session. So if you want, you can always look at the video, but Yura has also made notes. So thank you for that. Yeah. Yeah. So as we discussed last time, uh, making or sequencing RNA is a multiple step process in which you modify the RNA to quite a large amount. So adapters are added, Illumina barcodes are added, and all of these additions have to be taken care of when we want to count the original RNA molecules, as well as then quantify them between samples, right? So um, two of the most important things are the amount of RNA that you sample, because in this step, when you're ligating an adapter, maybe in some cases you get a lot of molecules, and in some cases you capture less molecules. So from sample to sample, you will have some differences, right? Um, and the second thing is, during this amplification step here, index addition, PCR amplification, at that time also it is possible that some molecules get amplified more than others, right? So there could be amplification biases. It's difficult to remove amplification biases in the way that we are doing it, but I will talk about it a little bit. Yeah. So the complete mapping pipeline, it's a multiple step process, and we went through it for a single end read last time, and today we will go through paired end reads. But essentially, what you start off with is a FASTQ file, um, um, which is here sequence reads, right? And the FASTQ file has the sequence as well as the quality scores for each base pair that is called, right? Then you can look at the quality of the sequencing. You can do adapter trimming, and it says here that it's optional. And we'll talk about when this is optional and when this is not optional. Yeah. Um, Next is mapping to genome. So last time we just mapped to the genome, but this time we will be actually using a splice aware mapping to genome. So this splice awareness is very important when you are doing RNA-seq, because you only get reads from exons, or most of the reads will be from exons, and exons may not be continuous on the genome, right? So they are separated by introns. So the mapping software that you are using has to understand that exons are not continuous. Yeah? So they have to be splice aware, right? And then you do counting, and then some statistical analysis. So this is what we will go through today. For we will not go through Go analysis, and also like making heat maps and everything. We will stop at this step. That's because there are better programs apart from Galaxy which are capable of doing that, and I will mention them on the GitHub page, and links for those. But until here, Galaxy is really good. Yeah. So we'll go through this kind of pipeline. So okay, so I think we will start, because we will start, and while the processing is going on in Galaxy, we can talk about these things like what is splice awareness and why is adapter trimming optional. Yeah. So first thing is we open the browser, right, and we go to the use Galaxy page, yeah, or the Galaxy platform, and here, if you have not logged in, then you can log in. Yeah. You don't need to log in to use Galaxy, but it's just that way you can save your history, you can save your workflows and all of this. Yeah. So once you are logged in, either you will have, if you have already used Galaxy before, then you will have some history from previous usage, or it may be empty. If it's not empty, then you can make a new history. So by clicking the plus button, which creates a new history, or you can also go to the view all histories page, and here you will have all of your previous histories, and here also you can generate a new history, so where it says create new, or you can go to one of your old histories by just clicking on this switch to button. Yeah. So we will use this history. We go back to analyze data, right, and we will name this history. So we will say RNA-Seq. So after you have typed something in here to name the history, press enter, because if you type something and if you click somewhere else, then it goes away. Yeah. So after typing the name here, just press enter. And next we have to get our files, the FastQ files. So for that, we can either directly upload or use the FTP server. So I will be using FTP, although the files also this time are not very big. So on the GitHub page, the files were zipped, so you will have to unzip them, and then you will get 6 files. So what you should have right now, if you don't have it, then there's just, you can pause, go on the GitHub website, and then you should have it. So what you should have right now are these 8 files. So wild type 1, wild type 1, R1 and R2, FastQ and gunzip. So .gz is gunzip, and it's an compression algorithm, but it's open source, and that is why most of the sequencing data is compressed using gunzip. Yeah, and FastQ means that it's just a FastQ format, right? So you should have these 8 files. Yeah, and on the GitHub page, it's this folder which is just zipped. So you just unzip it. Yeah. And then using your FTP client, we can connect. So because I'm using the European Galaxy, so use galaxy.eu, I will be connecting to the uni-Freiburg server, the Galaxy server at Freiburg University. Yeah. This is for whole of Europe, not just for Germany. Yeah, it's faster than the other server from ULB. Yeah, but if you're in US or Australia, you can use their other ones. I think it's just use galaxy.org for US. Yeah? And we will make the connection. Yeah. For making the connection, it's still username and password, the same one. Yeah? And we will put all the 8 files. Okay. Yeah. You just put them in the directory that opens up. You don't have to make a subdirectory. You can make a subdirectory, but you don't have to. Yeah. So you can just put all of them together. Yeah. And then we get them into Galaxy by saying get Galaxy, get data, and upload file from your computer. So here we could have just chosen the file, but then the transfer is using the HTTP protocol. So the protocol that's used to show this website. So this HTTP protocol, it's a bit unstable sometimes, because in the middle, especially if your files are like big files. So in this case, we only have 15,000 reads, but if you have a few million reads, then it could be that it breaks in the middle. FTP is just more stable. Yeah. So either you can do that for today, or go through the FTP. Yeah. And instead of taking all, we will just take 2. We will take the others later. Yeah. So or you can take all of them. It doesn't matter. Yeah. So I'll start this. Yeah. And now you see that these 2 files have appeared in my history. Right? So they are still gray, which means that this operation is in queue, and now they have turned yellow, orange with this rotating circle, which means that the process has started, and green means that they are now in Galaxy for use. Right? And you can view these files with the I button. So as you see, there is a header, right, which just has some ID for some information and an ID for every sequence. Then you have the sequence, then the plus sign, which denotes the end of the sequence, and then the quality scores. Yeah. So just like last time, the first thing to do is to trim. So we will be using trim galore. Where is it? Here, trim galore. It's in the. Why did I lose it again? Huh. Okay, I think I'll just go here. Yeah, so it's in the fasta/fastq directory, trim galore, right? And here, unlike last time, so last time we had a single end read. This time we had paired end. So paired end means that your 2 reads. So when you make your cDNA, you can either read it only from one end, or you can read them from both the ends. And paired end means they were read from both the ends. Yeah. And also in paired end, it's very essential that your reads are paired, right? So the first read here is the pair of the first read in the other file. Yeah. So that is why you cannot have unequal number of reads, right? Because every read should have a pair in the other one. Or even if they're unequal, then you have to specify which reads do not have a pair. Yeah. But typically you will have the exact same number of reads, right? Yeah. So that is why it's called paired end, right? And in trim galore, we choose paired end. Yeah. And then we put the 2 pairs. So the first one and the second one, and we say that automatically detect the adapter. And in the advanced settings, we ask it to generate a report file. This will just give you some statistics about how many sequences were trimmed, how many sequences passed the quality control, because trim adapter also does quality control in 2 different ways. So the first quality control is that here. Yes. So if the quality, the read quality or the base pair quality is less than 20, then it is removed, right? So this is for low quality bases. And after removing of adapter and removing of bad quality reads, If the length of the sequence is less than 20, then it's completely discarded. Yeah. And if for paired end reads, it will not only discard one of the pairs, but it will discard both of them if even one of them does not satisfy this condition. Um, there are options here, like for example, specify if you would like to retain unpaired reads, in which if one of them does not specify, does not clear the quality criteria, at least the other one is still, well, not kept in the same file, but it makes a new file. So it will make it will output actually 4 files: the 2 paired end trimmed files, and then 1 single ended file for which their corresponding pair was removed. Yeah, so that's why there will be 4 files. Okay? But we don't need that, because we will just deal with the paired ends, and this is the typical behavior that this is the typical pipeline that you use. You normally do not care about if one of the pair is not aligning, then the other one is also thrown out. Yeah. So just in the advanced parameter, we say yes, and all the other things we keep as default, right? And then we execute. So in execute, you see that you get 3 outputs. So the first 2 outputs are the trimmed FASTQ files, right? And the third output is the report file. So the report file will have some statistics about the trimming. And the next step is mapping, right? So we are here, we have trimmed the adapter, or at least, I mean, we have run the program to trim it. And next file is to map it to the genome. So for mapping it to the genome, there are 2 options, star or HiSat2. And we'll be using HiSat2. Yeah. Now for mapping to the genome, you have to know which organism or which animal did the samples come from. So they actually come from zebrafish. Yeah. So that's why we will be mapping them to the zebrafish genome. And so we run HiSat2. It's part of mapping. So these are just, I've put them in my favorites. If you do not have them in your favorites, you can always just search here. But for some reason, the search, yeah, it sometimes gives multiple ones, difficult to find. And if you like, if you like a tool and you are going to use it a lot, add it to the favorite by clicking here. Yeah. So we will be using the zebrafish genome, the most recent one, 11, right? And then we say that it's paired end, right? And as input, it will be the 2 paired end reads from trim galore at the output. So the trimmed read 1 and the trimmed read 2. And we leave everything as default. So our RNA library is unstranded. An unstranded library is typically made when we digest the RNA into smaller fragments. So like here. So if the RNA is fragmented and you use a random hexamer with adapters, then you can imagine that it can bind. I mean, your library does not really have an orientation, right? But if you use a poly T primers, right, then your library will have stranded, because it will only bind to the 3' end, yeah, and extend in the other direction, right? So then it becomes a stranded library. But in this case, it's unstranded. Most of the time, you can leave it unstranded unless and until you are really interested in some kind of information. You are interested in some specific information. You can leave it unstranded here. And then paired end options, we leave it as default, but we can check the options right now. Yeah. So, okay, we will run this program and then we will go through the options. But just we will say in the summary options, we will say print alignment summary. Yeah. So this will give you statistics about the alignment. And then in all of these, we do not. Yes. Sorry. There is one more alignment. There is one more parameter to set. Last time we had not done it, and the correct way for mapping RNA-Seq is to use splice, something that knows, that is splice aware. Yeah. So what does that mean? It means that you will have to tell the program where the exons are, right? Only then it will know where splicing can happen, right? And the mapping is done against a genome, right? So here it's using a genome. In the genome, there is no information about exons, right? The information about exons exist in GTF files, right, which contains genes and their exons. So the GTF files which were used for counting, just like in last session. Yeah. So we have to use the genome for mapping, and to make it splice aware. So HiSat2 is a splice aware mapping algorithm. We have to set the parameter here, spliced alignment options in advanced options. Yeah. So just to recap, we'll select the genome for zebrafish because the sequences come from zebrafish. We'll choose to map the two trimmed files, the read1 and read2. They are paired. And in the summary options, we'll say that print the alignment summary. And then in the advanced option, we'll make it splice aware. And how do we make it splice aware? We'll say that we want to specify splice awareness options, and here we'll have to provide it with the GTF file, right? Now, unlike the genomes, which are, and a lot of genomes are present in Galaxy by default, GTF files are not, right? So there is nothing. So the one thing that we have to do is we'll have to upload the GTF file. So that file that contains information about transcriptome, right? About genes and transcriptome. And this GTF file has to be from the same source as the genome file. And Galaxy is getting its genomes from UCSC. So the transcriptome file also has to come from UCSC, right? So last time we had downloaded it, but otherwise I can just show you very quickly. UCSC browser. Okay, we'll go here. We'll go to downloads, genome data, and then other vertebrates, zebrafish. And so this is the version that we are using, Danrer 11. You'll find it here. Yeah. Then Danurario 11. And then we go for sequence file and annotation. Here we go. So these are all the different variants of the genome. So this is the genome file, danrer.r.fa. So FASTA file, which has the full genome. Yeah. And these are just different formats in which the genome has been presented. Yeah. And the genes will have information about GTF file. So we can select any one. I'll just select the ref gene and we will download it. Documents. Yeah. And once it's downloaded, we will have to get the data in again. So this time I will just do via HTTP, not via the FTP server. It's not that big a file, so it should be fine. And then it will come. But the good thing about Galaxy is you don't have to wait for their things to finish. So here you can click on browse datasets, and even if it has not uploaded, you can select it. So right now it's unavailable, but when it becomes available, then it will be done. So with all of these parameters, we execute. Yeah. So now this thing is running, and while it is running, we'll discuss what is splice awareness. But I think the term is self-explanatory. So here are your pairs, the pair of reads that you have gotten from sequencing. So either they can lie on the genome, either they can completely lie on the genome right next to each other, separated by like continuously on the genome. Sorry. So you can have, so the sequence that you have is in red and is in black, and in red are the two adapters, right? And this complete sequence maps on the genome in continuity. But this may not be the case if the two sequences span an intron. So then there are multiple different possibilities. The two sequences might be in the same exon. Well, then on the genome they are fine because they are continuous on the genome. But it could be that they are that one of the reads is in one exon and the another read is on the other exon, right? Then they lie far away on the genome, or it could also be that within a read is where the splicing is happening, is the site of splicing. So half of the read lies in one exon, then it's split, and then the other half of the read lies in another exon, right? And then you get your other end of the sequence. Yeah. So this splice awareness is important because otherwise these things will be missed, because in this case when you make an alignment score, it will have very bad alignment score against genome because only half of it is aligning and the other half is technically not aligning because it's not a continuous region on the genome, right? So that is why you need this kind of splice awareness, and both star and HiSat2, these are splice aware, and most of the uh, mapping tools available nowadays are splice aware. But to make them splice aware, you need the genome as well as the transcriptome, right? Because the transcriptome will have information about exons and their position. Yeah? The genome doesn't have, the genome file doesn't have such an information, right? So that is what it means by splice awareness. Okay, so the HiSat2 is running. So we will let it run. Until then, we can see the report file for our trimming. Yeah. So this was the report file, and it auto detected this adapter sequence, right? And 703 bases out of 1.5 million base pairs were low quality, which were removed. Yeah, so it's very few. So most of them were high quality, right? And yeah, that's about it. Yeah, in this report that you are interested in. Yeah. Another parameter that you would be interested in is how many reads were finally written into the trimmed file. So here all 15,000 reads were written. So not even a single read was removed because it did not pass the quality control. So that means that all of them were good quality. And this is also true for the other pair. Yeah. So 15,000 sequences processed in total. Yeah. And 15,000 sequences processed and 15,000 pairs, sequence pair were analyzed. Okay, and number of sequence pair removed, because yes, here we have that information. So 31 pairs were removed because at least one of the read was shorter than 20 base pair, which was our cutoff. I will just zoom in. So our cutoff was 20 base pair. So 31 of them were shorter, and that's why 31 were removed. So let's see actually were they removed. So if you look at this file, it has 14,969 sequences, right? Which is 31 less than 15,000 sequences, which were in our original file. So in this one also, in the other one also, 14,969, and here 15,000, right? So 31 pairs did not pass quality control. Yeah. Okay. So while HiSat is running, we will go forward and start the counting, at least setting it up. So for counting, we will be using HT-Seq count. Yeah. And this is the counting algorithm. So it will count the number of reads belonging to a particular gene. Yeah, and it will aggregate all the information for all of its exons. So it will not count per exon, but for all of the genes, right? And so it needs the output of the genome mapping. Yeah, so which will be here once it's finished, and it will need the GTF file, right? So the GTF file will have information about the genes and exons. So these two, with these two information, it will be able to make the count. Only thing is that we have to change that our sequencing is not stranded. Yeah, and here this is default, but it only says that count the exons, right? And gene ID means, so count the reads belonging to exons, and second thing is aggregate them and give me one result for every gene ID. Yeah. So if you are interested in making counts for every exon, then this is the place to change from gene ID to exon. So whatever you specify here, your final result file will have results for that feature. So gene ID means for a unique gene ID, give me the counts. If you specify exons here, then it will mean for every unique exon, give me the counts. Yeah. And in the advanced options, again, we will go and we don't have to change anything. Yeah. And we can just execute. Right? And again, HTSeq by itself makes two files. So actually, we do not have to specify that we need a report. The second file is the report file. Yeah. So until then, we can see the summary of our mapping. So in the summary of our mapping, now this looks different than a single end read, right? It has many more parameters. Yeah, because it is going to tell you about how the mapping went for both the reads. Yeah. So here it says that 14,000, like out of the total 14,969 reads, 1,423 reads did not align. We'll go into what is concordantly. Yeah. And 74% of the reads aligned exactly one time, and 2,000 aligned more than one times. Yeah. And then out of the and this is about the pair. So if whether or not both the reads aligned zero one time or more than one time, right? And if the pair aligned 0 times, then it takes each read individually, so one of the two pairs, and then tries to align them again. So just to check if one of the two will align. Yeah. And when it does it, 713 of these align exactly one time. So it also keeps them in the final result table. And with this, the total alignment rate is 94%. So that's more than this, right? Because it's aligning the pairs and then individual ones which don't align. So what is concordantly and discordantly? That is in HiSat2. So if we go to HiSat2, it tells you about what do these things mean in the advanced options, I think. Just let me think. Where is it? Summary options? No. Yeah, here we will see it here in all of the options. Yes. So there in the paired end options, it explains what this means. So what it means is that if you have specified an orientation, then it should find the pairs in the same orientation, right? That is first thing. And the second thing is you also give it an average distance between the two pairs. So where is the paired end option? Yeah, we have, that is why I couldn't see it. We have to specify paired end reads, and then it gives you paired end options. Yes, all of these things is here. Yeah. So you have to specify how far away they are. I think typically it's like 200 or 300 base pairs, right? Because when you make cDNA, there is a size selection, right? So your the ends of the two reads cannot be more than the two reads cannot be more than 300 base pairs away. Yeah, but if they are away, then they are considered discordant. Yeah? So it will still consider them, but it will say that they are not concordant. So that means that they are behaving differently than what was expected. Yeah? So they are mapping and all of these kind of things are happening, but they are not proper, let's say. Yeah. It will also happen if you have a stranded library and the one of the end is downstream or upstream and all of these options, right? So you can go through this if you are interested, but most of the time, most of your reads will be concordant, and a few of them which are not, it can, I mean, they might also map to the genome, and this is completely fine. Yeah. Okay. Uh, cool. So now we have our counting done, and another thing to consider is that what I wanted to say is that your alignment rate is a good parameter to know how good your sequencing went. So this is one parameter to either keep in records or to compare between your sequencing runs, because something like less than 30%, it may be that there was some problem that happened in the pipeline during the preparation of RNA, or the amount of RNA you started with was very, very less. So most of the time, if you start with like RNA that is collected from a few cells, like single cell or even like 20, 100 cells, then your alignment rates are typically low. That's because there is a lot of primer dimer and there is a lot of amplification of incorrect reads. And because you have to amplify your cDNA a lot, so even mutations get included, mutations are introduced. So then the alignment rate decreases. But this is a number to keep in your record when you're comparing different ones. Anything above 60% is really good. Another thing is, if your alignment rate is really low, that also means that you are wasting a lot of your sequencing and your money on just sequencing crap, right? Because here only 7% of the reads are not useful. But let's say if your alignment rate was 20%, that means 80% of the reads were not useful to you. Yeah. So this is just a number to keep in mind. Sometimes even a low number is fine because you get enough reads to analyze. But uh, if you are consistently getting low numbers, then there is some problem with pipeline. Yeah. Anyway, so we have our counts, and we can also see the report of the counts. I mean, this report is not really that important, because all it says is that how many reads did not map, but it doesn't really say out of how many, but it will be out of 15,000. So out of all the reads that were in the BAM file, 3,000 were not found in exons. So that means that those reads might either be in introns or might either be in non-exonic regions, because we said to only consider exons. Right? And then we look at our counts table, and this is, it's taking its, it sometimes gets stuck here. It's pretty big, yeah. So here we will have all of our genes. Now this is the most important file, because this is a file that you will be using for analysis, right? And here in this file, you should have some non-zero reads, right? Because if it maps, then you should find some genes, right? So for this file, for this output of the HTC count, we will rename it and also put a tag. So I will just zoom in. Yeah, maybe that's too much. Okay. So first we can put a tag by putting this edit dataset tags, and we will say hashtag wild type _1 and then press enter. So you press the edit tag button, write your tag, typically with a hash, and then you can press enter, right? And this will add a tag. Also, we can change it. We can change the name. So if you press on this pencil mark, which is edit attributes, you can change the name of the file. And we'll change it to wild type _1 underscore. Well, okay, counts underscore wild type underscore. Yeah. And we save it. So because this is the file that we will be needing later. Because otherwise what happens is you always keep seeing on data 6, data 7, like it's the name of the data, and then after a while it becomes too confusing. So the next thing is that we can do this for the other 3 data sets, right? We have 1 more wild type and 2 more treatments. But that is a bit tedious, right? I mean, you have to sit down and run all of these things. And that's why Galaxy has a really nice feature, which is called workflow. And this is what we will be talking about next. Yeah. So how to make a workflow and how to use it. So what is workflow is it will automate all the steps of your pipeline. So you don't have to choose the tools to do this analysis. Yeah. And the way to make a workflow is that there are 2 ways. The first way, let's see the first way. So you can go to the workflow and let's add a new workflow. So you click on this workflow button and then you add, click the plus button, and we can name it something. RNA-Seq 1. And then you see an empty page. Yeah. And in this page, you can take any of the tools that they have mentioned, that they have present in analyzed data. So all of except upload file from your computer. So this is not a tool here, or it's not a template here, but it's replaced with input data set, because it doesn't really care how you input your data set, but it's just an input data, and you can drag and drop. No, you have to double click once on it, yeah, and you will get this kind of tools here, this kind of box. Yeah. So what you're trying to make is you're trying to make a pipeline, but it is in which you are going to connect your inputs to your outputs via some sort of analysis or some sort of manipulation. And in this, you can make the label. So you can say pair _1. So the first pair, and annotation can be fastq, or we can say even fastq.gz. Right? So this is our annotation, and then we say save. So the second data will be pair _2 and fastq.gz. Next, whenever we have our 2 data sets, so the pair 1 and pair 2, the first thing to do is to trim them, right, with trim galore. So we search for them and we click, right? So now you see that a new tool comes up and you can connect your input to your output, right? Sorry, this is not correct. Yeah. So you see that trim galore only has 1 input, right? But if we want to trim paired files, we have to give them as 2 inputs which are combined to the 2 trim galore, right? And so we have to select PairedEnd. So all the parameters related to a tool are on the right hand side, right? And if you change anything, then it also changes the tool here. So now we can connect the outputs, right? And I think you get the idea of how a workflow is working. Next, so this will be, so the input will be the two files that we have. They go connect here, and then it gives you two outputs, which are the two trimmed PairedEnd reads, right? So nothing is happening. I mean, we are just making a pipeline, theoretical pipeline, right? But later on, we can run it in one step. So if you just click on this button, then it will run, right? Then the next step is to do HP to map, and for that we use HiSat2. So HiSat2, we click on this tool, and again we have to go here and select PairedEnd, right? So in single end it only had one input. Now with PairedEnd it has two inputs, and we can also specify our genome here, zebrafish, right? And we also have to make it splice aware, right? So so to make it splice aware, we go to splice awareness options and specify splice alignment. So specify the GTF file, and now you see another input came for the GTF file, right? But we don't have a GTF file, right? So we'll have to put one more input because the GTF file is not present in Galaxy. It comes as another input, and here we can say GTF file, and the annotation will be, it will be GTF, right? So this will be the input here. Here we input, yep, right? And now we can select um, our our splice awareness. We can modify it. So where is splice awareness? Okay, we don't have to modify the input because that anyway goes as an input, right? And the output is a BAM file, right? And you will see that there is a star. So for every output you see a star, and if you click on it, it becomes fluorescent green in color. If an output is fluorescent green in color, only then when this workflow is run, you see it comes as an output. Otherwise, it runs in the background. So if you have a very big pipeline in which there are multiple outputs in the middle, but you're not interested in them, you're only interested in the final one, this is also very helpful because you can just hide them. So it makes your workflow much cleaner, right? So now we have done this. Now we have the BAM file and we want to make the counting. So we count them with HT-Seq count. So as always, the counting requires two files, an BAM file and a GTF file. For some reason, this tool calls it a GFF file, but it only accepts a GTF file. So this is something to be careful with because I have tried giving it a GFF file, which is another format for transcriptome data, but it actually fails. So I don't know why they don't call it that. And then you connect your output here and this input here, right? And you can also first save, right? And then you can also go here and you can say auto relayout, and it will make it a bit more cleaner, right? So your three inputs, your files are trimmed, and your GTF file goes as an input to the mapping as well as to the read counting. Yeah. And now you have your two outputs, which you want to see, right? Because these are something you are interested in. So we will put only those two as dark green, right? And we will save it. Yeah. So this is your entire workflow. It has been made, right? And now we can run it. So before we run it, we should actually upload our remaining files, right? So we go back to analyze data. and we say get data. Why does it say the history has been purged and deleted? That's weird. Anyway, okay, so we say get data and upload data from your computer. Choose FTP and I have no idea why it says this. I think this is a bug because clearly the history is present and it's available. So I think this is just a bug. Yeah. So now while this thing is going on, we can just go to our workflow and say that we want to run the one that we just made, right? Yeah. And now you see that it asks you if you want to send your result to a new history, but no, we'll keep it in the same history. And now you see that it gives you the 3 input options that we had specified. So the 2 reads. So in this case, it will be wild type 2 and the second read for wild type 2. And the GTF file will be this. So you can specify the other options here, but you don't have to because we have already set them up in our workflow. And you also see that all the 3 tools which will work on these input data sets are already specified here. So you don't have to do anything. And then you just say run this workflow. Yeah. And now you will also see that it's going to load all the steps. So not just one step, it's going to do all of them. And it's going to execute them, of course, one by one. And when the result appears, it will put the counts table on top here in your history. It will not show the other ones because we had hidden the other ones, right? So this is one way of making your workflow in which you actually pull things together. Yeah. Let us see if it's actually running the workflow, because for some reason I have no idea why it should have started it. I'm just trying to figure out why it has not yet started running the workflow. So I guess I'll just run it again just to be safe. And just to be a bit safe, I'll run it in a new workflow. Okay. I mean, the one problem is that yes, it finally successfully scheduled all the things. I think it did not, for some reason, I don't know why it did not do it the last time. So now all the steps have been scheduled, and it will say if it's created. But if we go to that history, we should see the steps being executed. Yeah, here. Now you see all the steps. At this stage, you see all the steps, but as they are getting executed, and if they were hidden, they will actually disappear and they'll go in the background. But initially, you see everything. Yeah. So this will be for that data set. Okay. So for some reason, yeah, the previous history just disappeared. Okay. I have no idea why, but anyway, we can make it again. Yeah. Okay. So this was one way of making workflow, right? And the other way of making a workflow is so you see that the two trimmed the two trimmings had happened, and therefore they disappeared because they were not part of our output. But so the other way of making a workflow is to go to this history and to go to history options and to say extract workflow. So if you have done multiple steps one by one, then you can also say to extract your workflow, and Galaxy will put all of those steps into a single workflow. So you don't have to make it like the way that we did. But the only thing is that you can extract a workflow only after all of your steps have been executed. So it cannot really extract workflow from unexecuted steps, right? So once all of these things are executed, I will extract the workflow because the other history disappeared somewhere, right? So anyway, I think we'll have to redo the first one again. So I'll upload the files here, and let's just upload all the four now. Close. Most possibly, I'll also have to upload the wild type one because its results disappeared. So I'll also do that. I'll just do it from the local file. Yeah. Okay. Good. So while they are uploading, we can run them. We can run the workflow already. So we will say to run this. And another advantage of having a workflow is you can get email many times. You can get email reminder if the workflow finished. Let me just see where that thing is. Workflow, edit workflow. Okay. Why is this thing starred? So when you, yes. So when you are in this workflow editor and you select a tool, then you can say email notification yes. This is only, this feature is only available when you make a workflow in this workflow editor. Yeah, it's not available for tools in general, but if you have a workflow, then you can tell it to email to notify you by email, and this email that it uses is the same that you used for login. So that would mean that when you have a workflow and you have started it, once it finishes, you will get an email that it has finished. So it's really, really useful because then once you start a workflow, it's on the Galaxy server, so you can close your tab, you can close Galaxy, you can even close your computer, and then once it's finished, you don't have to go to Galaxy to check if it's finishing. You just say email notification, and when it finishes, you get a notification email. um, you will get a notification if it's successful or even if it's unsuccessful. So if some error happened, you will get an email notification. So this is really helpful, and this feature is only available in this workflow planner. Yeah. So this is good. So I will say email notification yes, and I will save it. Yeah. And then we will run it again. And we will run it for um, the wild type one. Unfortunately, it went somewhere. I'll also have to upload a GTF file. Okay. Okay. Great. So you see that it actually scheduled the complete workflow even though the GTF file is still loading. That's because it will just wait for it to load and then run the remaining ones. So until that time, we can rename this file. So this is the count file that we got from wild type under to the second wild type sample or the second control sample. And we can just rename it to counts underscore wild type underscore true. We can say control. WT is just wild type. Yeah? Then we say save. If you want, you can also edit the attributes. So now we will see how to extract the workflow from here. So you can see that there are two hidden things. So the two hidden files were the trimmed files and the HiSat2 file. Yeah, actually there should have been three, but I think in the old workflow we did not we did not make it hidden. But in the next one it will be hidden, right? So you can see the hidden files from here, and let us extract the workflow. So we say extract workflow, and it will only extract from the files which has already been run, right? Yeah. So here it says some data sets still queued or running were ignored. So if a data set is queued or running, if it hasn't finished its run, then it won't become part of this workflow when you extract it. Yeah. So we can say workflow extracted, okay? And then you see that you can choose which part of all of this thing, all of the parameters that you have, you want them to be in your workflow. So all of these go out. Is there an uncheck all? Yeah, uncheck all, and then we say we only need these two. The advantage of extracting a workflow in this way is that the parameters that you had used to run these tools will also be saved. So you don't have to go into these tools and specify the parameters again. Yeah, so that's the good advantage of extracting them. Yeah. You will see that you do not see trimming here, and that's because trimming was hidden. So to also have it in the thing, we should unhide it, right? And then we will say again extract the workflow. Yeah, now you see trimming. Yeah, so if a data set is hidden or queued or running, then it doesn't become part of this extracted workflow. Then we say uncheck all, trimming, mapping, counting. Yeah. So we say create workflow, and we can go and look at it. So we can edit it. And I'll just say auto relayout and here. So you already see that you have your two reads, which is trimming, but there is no input, right? This is something that you just need to put. So you can just put it here, right? And connect it. Because there is no upload file from your computer as an input. It's just an input data set, right? So here we say read 1, read 2. And you will see that all the other parameters are already made. And we will just say that we are only interested in looking at this as an output. Sorry, one more input data set is remaining, the GTF1, the GTF, and this becomes the input for here and the input for here. Yeah. So this is another way in which you can make your workflow. Run it for one of the sample, and then once it's completely executed, then extract it. So these are very powerful way of doing it. And also for the last workflow, we go and say email notification, yes. And now you see that we did not actually specify any of the parameters because the parameters that we had used for running, they are present here. So we do not have to change them. Yeah. So we can save this. Okay. So this is more or less how you will get your counts file. And then next thing we will do is, okay, so now we have the next thing we will do is to make the differential gene expression analysis. But before doing that, I mean, we'll have to run the same workflow on treated samples. So first we got this data set, so we will also change its name. So it's the HTC count on 14 and 17. So 14 and 17 are hidden, and 17 is the mapping on 40 on data set 40 16 and 15, which is from 1 and 10. That's a bit weird. Let me just see the info. You can always go to info and see what the inputs were. I hope that we did not give it. Oh, no, it is wild type 1. Okay. Okay. So this is wild type 1. So that means that this HT-seq should be called counts underscore wild type 1 or control 1. And we can save it. So now we have the counts for control, the first control, we have the counts for the second control, right? And now we will make the counts for the 2 treatment samples, and we will do it via workflow. So we'll just say run, and we do it for the 2 treatments. So treat 1 and treat 1, yeah, and we run this workflow. Great. Let it just start scheduling before we change the page. Cool. So these things are scheduled and they will run. Okay. So while they are running, we'll discuss about why we talked earlier about why adapter trimming is an optional thing. It's not a necessary thing to do. And the reason why it's not a necessary thing to do is because the mapping tool that we are using, so both HiSat2 and STAR, at least these 2 mapping tools, they allow what is called as soft clipping. And what is soft clipping? So when these mapping software are aligning your reads to genome, as soon as they find a part of your read that maps to the genome, they will have a little bit of unmapped portion. Yeah. So if your full read maps to the genome, then all is good. Yeah. So then you can forget about it. Then it's perfect. But if there is only a partial match, then there are different scenarios which might happen. The first scenario is that the remaining part of the read maps to the genome, but at a different location in the exon. At a different location in the genome, but which is not continuous. So this is allowed if the mapping tool is splice aware, right? So this is what is splice awareness. Yeah. Next thing is you might have a mismatch. So maybe there was a mutation during sequencing, or there is a SNP, right? So the animals or the samples that you used had a SNP. They had a variation which was not in the reference genome, right? Because the reference genome comes from a different animal or samples. And if there is a mismatch, then these tools can ignore this mismatch to some extent and then continue. So they will continue to search after this mismatch. Yeah. And because of this quality, I mean, wherever there is a mismatch, the mapping score will decrease, right? But it will still continue. And because of this quality, even if your read has an adapter, it will not throw away this alignment. And this quality, this property of still keeping this is called soft clipping. So if you do not trim with the HiSat2 and STAR, it's most of the time completely fine. You can skip the trimming step. It will do what is called soft clipping, and soft clipping is essentially it puts a penalty on the amount of mismatches that you can have. A penalty on each mismatch you have, and it puts like a maximum penalty. And then it looks if your mapping score minus the penalty is still good enough, then it will keep it. And most of the time, the adapters don't really reduce your mapping score enough that it throws it away. So this soft clipping property means that you can ignore your mapping score. You can ignore trimming your data most of the time. Yeah. And therefore, most of the time, even if you have adapters or something, and if your tool is splice aware, then it will map it correctly. Yeah. So we can try that. Yeah. Okay, so this thing, the third thing is also finished. So just because it has finished, we will rename it as counts underscore treatment underscore one. And we'll save it. Then we'll have the workflow run again for the second treatment. Treat two, run the workflow. Okay. Typically, what I like to do, which I did not do today, is get two files. So, I mean, I put everything on the FTP server, but only bring one PairedEnd read into Galaxy, run the full workflow, then bring the next PairedEnd, then run the full workflow. So do it one by one. That's because then what happens is that you have your two reads and you have the results of those two reads right next to it. But here we got everything at once, because then it becomes confusing. You know, you have to always look back. Okay, this result is from this data, this data. But yes, sorry, I completely missed one step. So I'll just do one thing again. So one thing that you can do is that these tags are really helpful, because these tags pass from input to output. This is something that I missed. I apologize. So if you get a PairedEnd read, a good thing is to put a tag on it. So this we can play hash treat underscore two. Yeah. So this is one of the pairs of treatment underscore two, and we put a tag on it. We do this also for the next one. Yeah. So we go to edit dataset tag and we go treat underscore two. Yeah. And you will see that if your tag is exactly the same, one good thing is that the color of the tag is also remains the same. So it will be blue. So if you make a different tag, Galaxy colors them differently. Yeah. And now let us run the workflow on this. So we have tagged these two, so treat underscore two, and we run the workflow. And now the results will also get the same tag. And this way, it's a really nice way of following your data. So as soon as you upload your files into Galaxy, give them a tag. Do not change the name, but you can give them a tag. And these tags carry from input to output. Yeah. So we will run this workflow. Yeah. And until then, I mean, it was already run once, so we will change it. We'll change the name. But now you see, with the new workflow, all the outputs have this tag of treat underscore two. So you don't have to go back. Yeah. Sorry, I missed this initially. This makes life much more easier to trace back. Yeah. So read your raw data into Galaxy, give it a tag, and then run your workflow. Hide all the intermediate outputs. Just keep the final output, and you will have a really clean history in which you only see your inputs and the needed outputs, right? Okay. But while this workflow is running, at the end, we will also get these counts again with the tag of treatment underscore 2. But one thing we can do is first, we can try to, we can do the mapping without trimming. Yeah. And the other thing that we can also do is then start the differential gene expression. I think we have time, so we can first do the mapping without trimming. So we'll just go to the workflow and we will copy this. So we get a copy of this workflow, we will rename it, and we will say RNA-Seq, no trim. We will edit it. And in this workflow, we are going to delete our trimming. Put this directly as an input to the mapping tool and do auto layout. Yeah, let's just get, let's go to HiSat2 and also give us the alignment rates. Yeah. So we will say summary options, print alignment, and this we say we want to see. Yeah. So we will compare what percentage of reads align with and without trimming, and also what is the percentage of reads that map to exons with and without trimming. Yeah, so we will make this comparison. So we'll say save, and we will run this workflow. Let's run it on the second treatment because it's tagged, so we will know what the results are. Yeah. So let us run this on here, and we will say run workflow. I mean, in our case, it won't be that much because our files are really small, but when you have big files, not trimming can make things much faster without too much of a difference, to be honest. Most of the time, I do skip the trimming step because there is soft clipping. Just to show you soft clipping, I'm sorry that a lot of the time I'm jumping from one topic to another, but that's just because it's better to run these things and then discuss something else. Yeah. So where are these options for soft clipping? So if you go to HiSat2 or even to star, and if you go to advanced options, you will go to the scoring options. So specify scoring options. So here is where the soft clipping options are. Yeah, and this also is technically a soft clipping options. So here what it says is what is the penalty, the maximum penalty for soft clipping. So here, I mean, it calculates what was the mismatch and all these kind of things. It will calculate this and it will see what is the score. Yeah. And my mistake, I think soft clipping was not activated. We should have activated soft clipping in that thing. Okay. So we can see the output, and if it's not very good, then I thought that by default soft clipping was activated, but it seems it was not. So we can run HiSat2 again with soft clipping being activated. Yeah, but essentially these are all the options here related to soft clipping, and it's essentially a mathematical problem where you give a penalty for mismatch, but if the mapping is good enough, so your total score will be your mapped score minus the mismatch score, and if it's high enough, then it will still keep the read. Yeah. But we will see the results file. Yeah. So let us see the results. So this was actually the alignment with HiSat2 without the adapters being removed. and you see that it has almost 93% alignment rate, right? Which is not that different. We can go and check what was the alignment rate after trimming. So this was the trim galore, the trimming of treatment 2, and this was the HiSat2 that was run on the trimmed data. Yeah. So if we look at the trimmed data, sorry. This was run on untrimmed, and then the trimming data, I don't see the statistics. Maybe we ran it without asking for statistics, I guess. Another way to look at it is just look at the size of the BAM files, because the size of the BAM files correlate with how much data is in the BAM file, right? So without trimming, the size of the BAM file is 2.3 MB, and with trimming, the size of the BAM file is also 2.3 MB. Yeah. So it's not too much. And even though it doesn't really show a lot of the options here, yeah, you can actually see the results here. So here, 75.97% of the reads aligned perfectly fine, right? And then without trimming, it was 72% of the reads. But that lower percent could also be because a few low quality reads were removed during trimming, right? Which was not removed. Yeah. So it's not that dramatic a difference. I mean, 73% versus 76%. Yeah. But it saves you a few hours sometime if you don't trim your data. Yeah. Okay, so that is a thing. But I think again, I think we made a mistake in which we ran this HiSat2 without having the soft clip option turned on. So we will go here. We will make the edit. Yeah, and we will go to HiSat2. Yeah, and here you go to advanced options, scoring options, specify scoring options, and allow soft clipping. Yes, then we save it, right? So now with this workflow, it's perfectly fine. It's a little bit faster workflow, but it will give you your count data in just 2 steps, right? So you directly go from your data to mapping, and from mapping to counting. Yeah. It works most of the time. If you think that your sample, your data is being affected by not trimming, then do it. It's just a matter of taking more time. Yeah. But with these new mapping software tools, it's not something necessary. Okay. So so now we can go to differential gene expression analysis, and it's super easy. That's why I took a bit more time discussing all of these details. So for differential gene expression analysis, we use this tool called DE-Seq2, differential expression uh, uh, determines differentially expressed genes, right? It's called DE-Seq2. Yeah, and it's something that we'll be using a lot. So it will take counts data from different outputs. So you can either take it from HTC count or feature counts or string tie. So these are just the different tools which will do the counting. We used HTC count, but you could as well use feature counts. It's not very different. Only thing is that HTC count, you have to specify that it has no header. Yeah. But files with no header can be input file, right? So we have to turn that thing off. We have to remember to turn it off. Yeah. So no. Files have header? No. Yeah. Okay. So when we do differential gene expression, we want to compare some condition to something else, right? So that's what we want to do. We want to compare and then see which genes are differentially expressed. the parameter that you are going to compare is called factor. Yeah. So you specify what is your factor. So let's say we say that a factor is treatment. You can specify anything like some kind of treatment. Yeah. And then you can specify different levels of factors. So different level will mean different groupings. So this factor level is the groupings. So what are the different groups within this comparison? And what are the files that belong to those groups, right? So we have two groups. One of those is control, right? And the control files are counts and this. Sorry, this one and this one. I had to press my command button in on Mac to select the two, but maybe you can also do it with Shift. Yes, you can also try it. No, not with Shift. So with Control in Windows, maybe with Control to select the two files. And then which is your another group. So within this treatment, the two levels or the two groups are one is control and one is treat. And then we select our two treatment files. Yeah. And we say that our data is count data, right? We have not yet normalized it. Yeah. And we would want to have a normalized count. So normalizing means that you normalize it because each sample will have different levels of RNA molecules, right? Well, I mean, different levels or different number of RNA molecules that were sequenced, right? So, so to normalize for that or to compare between two samples, we normalize, so we get rid of the depth of sequencing. Yeah. So, and we can leave everything out, as it is, and we can say to run it, right? So while it is running, we can discuss another topic, which is that you see we had 8 samples, 2 paired end reads for each sample, right? And we were doing it one by one. Yeah, so we were taking one input, running a workflow or something. I mean, workflow already automates it, but still we had to do it one by one. Is there a simpler way in which we can put a sample name, put the two files, the two paired end files, but treat them as one, like because they are paired, so you can treat them as a combination? And this is also possible, and this you can do by making what is called a collection. Yeah. So let me just see. Yes. Dataset action. Yes. I just have to check where do we make the collections from. Hiding in datasets. Okay. Let us check here, collection. Structure, copy or publish. Okay, I'll have to check my notes until then. we can just wait for the DE-seq things to finish. Yeah, but that is the last topic to discuss today, and if I cannot do it, we'll discuss it next time. Yeah, because that is helpful. Okay, I'll just check quickly on Galaxy how to generate a collection for PairedEnd reads. Ah, yes, sorry. Perfect. Yes, now I remember. So you have to press on this tick mark, which says operations on multiple data sets. Yeah. And just select your inputs, right? So you can say which kind of inputs, which kind of data sets will become part of this collection. So you can just select your inputs. It's better to do it when you have just uploaded all your files. So let's actually do that. Let's make a new history because it will be much faster. Yeah. So we make a new history and we say RNA-Seq collections. Yeah. And now we will get our data. Yeah. So and instead of getting the data again from the computer, we will just copy and paste it. So this is the data that we want. We will put it here. We will put this thing here. Okay. Treatment. So this is how your history will look when you just start your analysis, if you upload everything. So one of the good things to do is to already put some tag on it, but it's not needed when you're making a collection. Yeah. And then we will press on this tick mark, which says operation on multiple data sets, and we'll select all. Yeah. And we'll say that for all that are selected, build a list of dataset pairs. Yeah. And now you see that it has already built your list, which is wrong, right? Because wild type 1 is paired with wild type 1 read 2 is paired with wild type 2 read 2, right? Yeah. I hope that this was okay what was being done. So I will just cancel it and do it again. So you may have all your inputs. You click on the check mark, select everything, and say build list of dataset pairs, right? And it will try to pair them together. Instead, so because our pairing, what it guesses is wrong, so you could say what is the thing that specifies your we can say unpair all, and then you can say what specifies your first read and what specifies your second read. So for us, all the first reads are specified by underscore R1 and all the second reads are specified by underscore R2, right? And then you can say pair this data set. And now it is paired. So all of your 4 data sets are paired. So now you can treat, when you do any manipulation on this one data set, it will actually automatically use both of them, right? And we say create a list. Okay, so we will say experiment. You can call it any other experiment. So we say create list. Right? And this has created a list of your 4 data sets, right? And now on to this, we can run our function. So let's say we run htseq2. Sorry, not htseq, HiSat. So we want to map. We directly want to go for mapping, not trimming, right? And we say that we want to use zebrafish, and they are paired end. and they are PairedEnd dataset collection. And now instead of specifying each of the PairedEnd, you just specify the collection, and it will run it for each and every one of them. Yeah. This you can also do in a workflow, right? So we can also go to a workflow and do this. So instead of input being a file, your input can be a data set collection. Yeah, so I will just do it here, then go to the workflow and show how to do it. And then we can change all the other options as you need. I will not run it because we already have run it, but this is just another way of making your life simpler, making your data set run faster and be easier to handle, right? Because now you do not have two files, you just have one. Yeah. And when you click on one of them, you have your forward and reverse, right? Yeah. So how do we change this in the workflow? We will see, but before that, let us look at the results of DE-Seq because it had stopped running. So I will go here and we will look at the results of DE-Seq too. So the result file is actually a PDF file and also a table file. So one thing you can do is you can download your normalized counts. So these normalized counts will be the counts on which analysis was done, right? So these are the counts after normalization. Yeah. So you will see that these counts might differ from your actual counts because they have been normalized, and they have been normalized to remove the effect of library size, which is the number of reads present in each sample. So in each sample, you might have different number of RNA molecules being sequenced, and this takes care of that. Then you can get your data, your result file, which will be the first file present. And here for each gene, you will have a fold change. So log2 fold change, right? And then you will have a p-value and also a p-adjusted. So here you will have the data for all the genes. You can download this and do some manipulation on Excel. So you can do, you can select only genes which have a higher p-value or a p-value less than 0.05. We can also do it in Galaxy, but I mean, you can use any tool that you want. Yeah? Okay. I mean, we only had 15,000 genes, so that is why you see a lot of non-applicable, and that's because it did not get any fold change for them. Yeah, that's only because we have 15,000 reads. Yeah? But normally you will have some p-value for all the genes. But the most important file is this PDF. I mean, your differential expression gene is one file, but this file is also very important because in here, what you see is your principal component, uh, principal components, and this shows how much is the variability between your samples. Yeah, and also how the samples are connected to each other. I have a feeling that I have mislabeled one of these files. Maybe, I don't know, but maybe when you run on it, it looks a little bit different, but anyhow. But here, what you see is that if you make the comparison here, your two samples, which are the control, are not very close to each other. So there is a lot of difference between them. And this is also true for the two treatment samples, right? So there is some difference between the control and treatment samples, but also there is a lot of difference between the two control samples. And this is because these samples were sequenced on different days. And because of that, I mean, either they were sequenced on different days, or maybe because they come from different animals or something like that. But technically, we should not have much difference between the control samples, right? So this difference maybe is removing or is hiding a lot of the difference in which we are interested in. So we are interested in comparing the control versus the treatment, but because of this variability, we might be losing a lot of that information. Yeah. And my feeling is that actually this is the treatment 2 and this is the treatment 1, and I have mislabeled them. Yeah. That's because originally the treatment 1 is close to control 1 and the treatment 2 is control 2 to control 2. Either I mislabeled them or because it's only 15,000 sequences, so the principal component doesn't look correct. Yeah. But what I'm trying to say is that because of this variability, it's something that is a batch effect. It's something that is a difference that you're not interested in. It's technical variability. So the biological variability that you are interested in is the variability between control and treatment, and not the variability between the control samples, right? Now this has not been taken care of when we ran this DE-seq2, and that's why we can go and take care of it now. So we would want to remove the effect of batch. So effect of having two different samples, so effect of having differences between the samples, yeah, and only keep the difference that is between control and treatment. So we'll go and run DE-seq2 again, yeah, and in this case, so factor is what defines the differences, right? But you can also include multiple factors, and if you do that, then it defines here. So you can input secondary factors that would influence your experiment, that might influence your experiment, that you're not really interested in. So like one of the factor could be the day that you collected your samples, right? It could be the chemistry, yeah. So maybe some of your samples are single-ended, some of your samples are paired-ended. Maybe they come from completely different sequencing reactions. Maybe they come from different gender. So if you have all of this information, you can specify them and also control for them. So all of these parameters, it will try to remove as much as possible, right? So the first factor that you specify is the factor for which you will get differential gene expression. And any factor that you specify after that becomes a secondary factor, and that becomes a covariate. Covariates are factors that might influence your experiment, but these are the factors you're not interested in. You are not interested in understanding, you're not interested in getting the variability from these things. You want to remove the variability, right? And that we do by adding another factor. Yeah, so by adding a secondary factor. So the first one is the most important one, the one for in which you're interested, and after that it's all that you're not interested in. Yeah, so this is one way of getting rid of your variability. Yeah, so we'll just do it again. So the first factor, we call it treatment, and this has two levels. One is treated, and we choose the two counts for treatment. Yeah, the next one is control, and we choose the two counts for control. Yeah, so this is the factor that we are interested in. We are interested in understanding what genes are changing because of treatment. And our two samples which were treated are these, and the two samples that were controls are these, right? But we think that there is a batch effect because these samples were collected on different days. Yeah. So one batch is batch 1, right? And on batch 1, we had collected the first control and the first treatment, right? So this is the first batch, and the second level in this factor is the second batch, which has the second control and the second treatment, right? So we are not interested in what is the difference between our first set of samples and our second set of samples. To go into a bit more history, actually the first control and the first treatment comes from cell which I think are collected from the same animal. I mean, actually I'm not really sure, but anyway, but this batch can also happen if you collect cells. Let's say you have a GFP positive population and a GFP negative population, right? And within the same within the same sample, right? So if you are interested in what is the difference between GFP positive and GFP negative within a sample, but you're not interested in differences between the samples, then you can also label each sample as a batch, right? Because it could be that some samples are at a different stage, or they come from a different passage, or the animals were born on different days or something like that, right? So all of these things you can just consider them as batch and you want to remove them. Yeah. And we say no because it's HT-seq, and we will just say output normalized data. Yeah. And then we can run it again. Yeah. I mean, in this case, we will not see much of a difference. The PCA plot will exactly look the same because the PCA plot is done on normal data. But when you have more than 15,000 reads and when you have an actual experiment, what you would see is that your differential expression, your genes which are differentially expressed would change, and some of them might actually change drastically. Yeah, so it's really important to look at the principal component and to try to see if there is a batch effect, and if there is a batch effect, to try to remove it. Yeah, using this. Another thing about principal component plots is that each axis of principal component defines a variance, and this means how much of the data variability is explained by points which are separated on that axis. Yeah, and one thing to remember about principal component is that the first component explains the highest amount of variability. Then you go to the second, which explains always less than the first, then the third, which will be less than second. So each component explains less and less and less variability. Yeah, and also in a plot, what is on the x-axis will explain more variability than what is on the y-axis. That's because on the x-axis is for the first component versus on the y-axis, which is second component. Yeah. So what it means is that so this axis explains 72% of variability. So it's divided into 1, 2, 3, 4, 5 parts, right? So that's approximately 14%. So from 0 to 0.1, this is 14% variability in your sample, right? But on the y-axis, it only explains 20% of your variability and is divided into 2 parts, well, more or less 3, so it's 6%. So on the y-axis, going from 0 to 0.1, this is only 6% of variability. Yeah. So that means that the scale is not the same for each principal component axis, and this can be quite misleading. So for example, if you look at this sample control 2 versus treatment 1, right? It's the total amount of difference between these two samples is high than the difference between treatment 1 and treatment 2. I mean, this difference is very, very low. It's only 6% of the difference. That's because it's also only the difference on this axis, which explains less amount of variability. Yeah. I mean, in other way to say this is that the variability that you see on the y-axis is going to be much lower than if points are separated on the x-axis. So separation on the x-axis in this case is 3 times, more than 3 times more stronger. Yeah. And this is something that people never consider, or it's not considered frequently when you look at principal components. So each axis does not explain the same amount of variability, and it also has different amount of strengths. And that's why also when you publish a principal component or when you show it, it's good to say how much variability it explains. That's because it could be like 90 and 10, and then the strength of difference on the y-axis could be very, very, very less. It could be minor. Yeah? So also in this case here. Okay, for some reason we got an error with this thing. Yeah, most possibly I think I have labeled my labeled the samples incorrectly, but I hope that you can run it, or if there is an error while you run it, then you can let us know. Yeah? But that's about it. That's all that I wanted to cover. And last thing, yes, so we go to our workflow, and within this workflow we can change our input data sets to input collection. So this we will do an edit here, right? And here, instead of this, we will put an input data set collection. And this collection operations, you'll have to split. Actually, it's better to run it. Yeah, I'll leave it. It's better to make the workflow from a run command. Okay. We will switch to this one. And we will say analyze data. Yeah. And then we go to HiSat2. Let's try to do this. So we will say zebrafish and PairedEnd collection, experiment, unstranded, print alignment, and advanced options. So important to have it splice aware. That is one thing, and also important to have it um, to allow soft clipping, right? Because we have not trimmed. So we will have to go for PairedEnd parameters and say allow soft clipping. Oh. Sorry, in scoring options. So allow soft clipping, yes. And then spliced alignment, specify this. Okay, no GTF file available, yes. I know. In data libraries, oh, back. Okay, we will put it from another history. This one, we move it here. Then we go to analyze. I mean, this is the only thing that we will just run, and then it will be. It's all for this session. As I said, we will not go into how to do Go analysis. I'll just put the links on some of the tools which is possible to do, where it's possible to do. But that's about it. Yeah. So summary options, we do this. Scoring option. Allow soft clipping and then splice rates. Yep. Okay, and then output options, align reads, no. Okay, that's it. So with this, you see that we did not have to run this 4 times. We just made a input collection and we ran it once, and it would make all the 4 together. Not only will it make all the 4 together, but it's also going to package them as one item. Yeah. And because of this kind of packaging, you get your data together. Yeah. So this is pretty nice, because in this case you do not you are not running the same thing again and again and again. It's much more automated, right? And everything runs together. And once you have run this one time, then just save it as a workflow. And then for every other experiment that you do, you can do this. Get an email when it's collected, when it's finished, and it's super automated, super quick, and really nice, helpful. Yeah. So on this collection itself, you can now also run HT-Seq. No SAM or BAM file available, yes. But we can do this. Sorry, we will say collection. I think it will have to wait for it to be either collection dataset, or we can say, we can call it on this. Yeah. All the four together. Yeah. And then we just let it execute. Yeah. Okay. So that's it. And then next week we will discuss how to do ATAC-seq, but it's pretty straightforward. I mean, once you are used to Galaxy, you can just search Google and search online which tools to use, and you just have to put the tools together. But we will just discuss a few intricacies of ATAC-seq and how it differs from RNA-seq. Okay, thank you, and have a nice weekend.